Welcome to Night Talks, where medical biology is in action. Ever had that feeling that someone's secretly communicating right under your nose? Like a group, plotting something, passing messages, waiting for the right moment to act? Well, guess what? Bacteria do that too. And don't be fooled, folks. They might be even better at it than we are. Bacteria can communicate, coordinate behaviors, and even build a protective, self-sustaining community. Not through words, but through a series of chemical communication systems. This ability allows bacteria to form biofilms. Come on, and let's take a closer look. So, what exactly is a biofilm? Think of it as highly structured bacterial community, similar to a fortified city, sort of a town with strong defenses like walls for enhanced protection from invaders. These bacteria adhere to surfaces and produce a slimy glue-like matrix called extracellular polymeric substances, or EPS. EPS consists of polysaccharides, proteins, extracellular DNA, or eDNA, and lipids. This protective barrier, once established, make bacteria harder to eliminate and is responsible for 60 to 80% of microbial infections, particularly those linked to medical devices like catheters and implants. But how do bacteria coordinate such well-organized defenses? Well, for them too, communication is the key. Although they have a fancier term than us, it is called quorum sensing. Quorum sensing is a chemical communication system where bacteria release signaling molecules called autoinducers to monitor their population density and coordinate group behaviors. When enough bacteria are present, they collectively activate genes responsible for biofilm formation. Simply put this way, it's bacteria using chemical signals to communicate with each other. When there are only few bacteria, the signals are weak. But when they reach a critical number, a quorum, they activate group behaviors to accomplish the collective task of forming biofilms. Well, honestly, they'd ace any group project, but in a way that's bad for us. Let's get serious. One prime culprit is Pseudomonas aeruginosa, a gram-negative opportunistic bacterium notorious for causing persistent nosocomial infections. Take, for instance, the catheter-associated urinary tract infection. Its QS ability enabled biofilm formation, making it highly resistant to a wide range of antibiotics and driving the development of multidrug resistance, which poses severe complications, particularly for immunocompromised individuals, including chemotherapy patients, organ transplant recipients, and people with HIV and AIDS. Biofilm formation is a multi-step process, allowing bacteria to establish a highly organized and protective community. Now, let's break it down step by step. Stage 1. Initial attachment. Free-floating or planktonic bacteria encounter a surface such as medical implants, catheters, or tissue in the body. In the early phase, the attachment is weak and reversible. Bacteria can still detach and move away. Stage 2. Aggregation and stable attachment. If conditions are favorable, bacteria strengthen their hold by producing extracellular polymeric substances, a sticky matrix that helps secure them in place. This marks the irreversible attachment phase, where bacteria commit to a biofilm formation. Stage 3. Microcolony formation. The attached bacteria multiply and cluster together, forming microcolonies. These microcolonies are the foundation of the biofilm, developing a distinct structure and composition. Stage 4. Maturation. The bacterial community grows, creating structured microenvironments within the biofilm, including oxygen-deprived zones that promote antibiotic resistance. Stage 5. Dispersal. Some bacteria break away from the biofilm and revert to their planktonic form, spreading to colonize new surfaces and restart the biofilm cycle. This isn't just a minor issue. According to the World Health Organization, antimicrobial resistance is one of the top 3 global health threats. Additionally, in July 2024, the Centers for Disease Control and Prevention classified P. aeruginosa as a serious global health threat due to its acquired multidrug resistance. Biofilm contributes significantly to this issue, as bacteria inside biofilms are up to 1,000 times more resistant to antimicrobial drugs than free-floating bacteria. Come to think of it, a fully armored and shielded formation, like the ancient Roman testudo formation or the Greek phalanx, is far more effective than a scattered soldier with no coordination. When individuals stand together, supporting and protecting one another, they become an impenetrable force rather than easy targets. Perfectly makes sense, right? But sounds like a serious problem, especially in the context of bacterial infection. To make matters worse, P. aeruginosa has a cunning survival trick called adaptive resistance, a temporary defense mechanism that significantly contributes to its multidrug resistance. But folks, this is not just a random occurrence, but a part of coordinated response triggered by quorum sensing alongside biofilm formation, where these microbes communicate and act together. Gram-negative bacteria like P. aeruginosa have an outer membrane that acts as a selective barrier. To allow nutrients in, they have porins. Normally, these porins also let antibiotics pass through. But when bacteria detects antibiotics, they reduce or modify porin channels, making it harder for drug to enter the cell. It means fewer antibiotic insides equal better survival. At the same time, it activates efflux pumps to push antibiotics out and modifies its metabolism to weaken the drug's effect. But here's the catch. This resistance isn't permanent. Once the antibiotics are gone, the bacteria switch back to normal, making infections recurrent and harder to treat. This cycle of evasion forces physicians to use stronger antibiotics or combination of therapies to deal with the infections. And if treatments keep failing, what options do we have left? The good news? Scientists are actively developing new strategies to counteract biofilm-related antibiotic resistance. One promising approach is quorum quenching, or QQ, a strategy that disrupts bacterial communication, stopping them from coordinating activities like biofilm formation and virulence. Instead of killing the bacteria directly, QQ block their signals, making them weaker and more vulnerable to treatment. Even better, because quorum quenching does not exert strong selective pressure like traditional antibiotics, bacteria are less likely to develop resistance. Thus, it is a promising alternative or complementary therapeutic avenue. To recap, bacteria use quorum sensing to communicate and coordinate behaviors like biofilm formation, which significantly enhances the resistance to traditional drugs and antibiotics, leading to multidrug resistant bacteria. Pseudomonas aeruginosa, a prime example, utilizes this mechanism to form resilient biofilms, making infections harder to treat. However, innovative scientific research strategies like quorum quenching offer hope by disrupting bacterial communication system. So, what's the point of all this? Why does understanding bacterial communication and biofilm resistance matter? Folks, because it's the key to developing innovative solutions against multi-drug resistant infection. The more we know, the better we can develop solutions. So, stay informed, support research, and share this knowledge. Together, we can outsmart bacteria. So, if you find this insightful, spread awareness by sharing this video. Thank you and see you next time.